As people are coming in, I want to welcome you and happy Thanksgiving to everybody. It's my pleasure and honor today to introduce our Grand Round speaker, Dr. Gail Pearson. Gail is a rarity. She's a native Washingtonian. Like me, there's only a few of us that can claim that fact. But she has other things I'd like to tell you about in her resume. She was recently promoted to adjunct professor here at George Washington University. Gail is director of National Heart, Lung, and Blood Institute's Adult and Pediatric Cardiac Research Program and is responsible for about $750 million in a research portfolio, give or take, depending on who's in the government. And she is in charge of the important Bench to Bassinet program, which I'm sure we'll hear about today, which includes the Pediatric Heart Network. And most importantly, she is a cardiologist here at Children's National, is one of our great educators and colleagues. So thank you, Gail, for presenting this today.
Thank you, Dr. Barul, for that kind introduction, the opportunity to be here today. In thinking about this presentation, I realized that my affiliation with Children's actually goes back farther than my CV would indicate. Like many members of my third grade class, I had my tonsils out at the old Children's Hospital building here on 13th and W Streets. This is how it looked in the late 1800s when it was first built, and a few years later when I had my tonsils out, I think it probably had changed a little bit. When Dr. Newman gave his grand rounds here a few months ago, he talked a lot about passion. So today I want to talk with you about something that I'm really passionate about, which is raising public awareness for the importance of pediatric research. Because as we all know, children are not little adults, but all too often the care we have to offer them is in the form of hand-me-downs from adult medicine. But first there's a quiz. So I'm not going to actually call on anybody, but I'd like you to raise your hand if you think you know the answer to this question. What is the name of the government agency that funds most of the medical research paid for by taxpayers in this country? I promise I won't call on anybody. Come on. Okay, good. Well, like people in this room, most Americans, if you ask them, agree about the importance of research. But in this poll that was done on behalf of Research America in 2010, it turns out that only 9% of people named the National Institutes of Health as the nation's leading biomedical research funder. Half of the people didn't have any idea, and it appears that the Food and Drug Administration, which got 20% of the vote, has a better marketing plan than NIH does. And furthermore, if you ask Americans to name a living scientist, they can't do it. If you just look at the person next to you, you know a living scientist. But for most Americans, the process and conduct of research is invisible. In fact, until a couple of years ago, this poll is conducted regularly. People mentioned Albert Einstein, who died actually in 1955. So I'm going to ask you today to join with me in helping to educate the other 91% of Americans who don't understand about the importance of research. And to do that, I'm just going to spend a few minutes on the National Institutes of Health and my institute, the National Heart, Lung, and Blood Institute, to give you the tools to help with this educational mission. So this is a picture on the left of Franklin Roosevelt dedicating the Bethesda campus of the NIH in 1940. Now, the history of NIH actually goes back to the late 18th century, but that's a story for another day. And at the time, President Roosevelt was standing on the steps of Building 1, shown there on the right, which is at the center of the Bethesda campus and currently where Francis Collins, the current director of NIH, has his offices. At that time, it was really the National Institute of Health. There really was only the National Cancer Institute. Today, there are 27 institutes and centers, and of course, the most important one is right there in the center in red. Some of you probably are also familiar with the Child Health Institute, which funds a lot of research here, the Clinical Center, and some of the other institutes. So the NIH budget's about $30 billion, give or take, and as Dr. Barul alluded to, there may be more take than give in the next few years. And nearly 90% of that budget is spent funding research at institutions like this one around the country and elsewhere. And roughly 10% is spent on intramural research, the research done in NIH's own labs. Now, NHLBI is the third largest institute. Our budget's about $3 billion. And we use this in roughly the same proportions to support research in heart, lung, and blood diseases, as well as training of investigators. So the main purpose of my talk today is to talk about pediatric research, as I mentioned. I'm going to spend most of the rest of the time on why we should do research in kids, and then talk a little bit about why we don't do more. And then I'm going to close with what the children themselves have to say. One of the reasons to do pediatric research is that they have diseases adults don't have. And so I think most of you will recognize this polio ward. It's true, of course, that adults, like President Roosevelt, can get polio. But in the middle of the last century, the main risk group was children aged 5 to 9. Now, it was unfortunate for President Roosevelt that he had polio, but he was from a very wealthy family, and his wealthy friends banded together to found the National Foundation for Infantile Paralysis. Today we know this as the March of Dimes. Polio peaked in about 1952 when there were 58,000 cases. People were terrified. They didn't want to send their children out. It would strike without warning. And along about the same time, Salk and others had been developing a vaccine. And in February 1954, they launched the largest human experiment in history. This involved 1.8 million children. 750,000 of them were in a placebo-controlled trial, and another approximately 1 million were in an observational study. This involved 44 school districts in 44 states in the country. In the observational study, second graders received the vaccine, and first and third graders were the observational cohort. This also involved nearly 300,000 volunteers, and amazingly enough, they enrolled nearly 2 million children and reported the results 14 months later, which happened to coincide with the 10th anniversary of Roosevelt's death. And it was clear from this study that the vaccine worked. The incidence of paralytic polio decreased by about 70%. And by 1979, there were no more cases of wild-type polio in this country. This is an extraordinary story. And I want to give you a little bit more of the backstory because I've been fascinated digging into this. So the main results of this trial were reported in Ann Arbor at the University of Michigan because Thomas Francis was the statistician and trialist who led the study. And this is the report. This report has no authors on it. It's 114 pages long. Today, the New England Journal gives you 2,500 words. This was announced not at a scientific meeting, but at a press conference. Now, the country had recently been on a wartime footing and really kind of following in that model, embraced this trial, and the children were called polio pioneers, everyone who got vaccinated. And the main dissemination plan, vaccinate Elvis. They figured that at least this would get teenage girls to be vaccinated. But in fact, by the end of 1955, the same year that the results were reported, more than 10 million children had been vaccinated. Now, this trial cost about $17 million, less than $10 a child, and was funded entirely through public funds, including $1 million in dimes collected from the March of Dimes. And for those of you who think you have trouble with data analysis now, for every one of those 1.8 million children, there was an IBM punch card at the Data Coordinating Center. So they were analyzing data on 1.8 million IBM punch cards. Continue with the theme of we should do pediatric research because they have diseases adults don't have. Birth defects are another condition. And again, of course, you can grow up to be an adult with a birth defect, but the chief problems really occur in childhood. This is Eileen Saxon. She has congenital heart disease, which is the most common structural birth defect. And she had the most common cyanotic congenital heart disease, which is tetralogy of Fallot. I'm sure most of you are familiar with this, but basically there's obstruction to the flow of blood going to the lungs. The deoxygenated blood instead goes to the left side of the heart. Children are blue. And Eileen, who at 15 months weighed approximately what a newborn would weigh, was moribund. And about a decade before the polio story, she became a pioneer of another kind, because Vivian Thomas, Alfred Blaylock, and Helen Taussig had been collaborating to develop a procedure that would alleviate cyanosis. And in fact, Eileen Saxon was the first human subject to undergo the blue baby operation. And this is shown here in this operating theater at Hopkins. So Eileen went to sleep in this operating room, a moribund blue baby, and woke up a pink pioneer in cardiovascular research. And this wasn't just a red letter day for pediatric cardiology. In fact, this was an instance where advances in pediatric medicine really opened the doors for much of adult cardiothoracic surgery as well. So when this was done, you didn't need a clinical trial to tell you that this was the right thing to do. The effect size was huge, and it clearly wasn't random. But in the intervening years, there have been so many advances that really now we need systematic evaluation of treatment strategies in congenital heart disease. So the Pediatric Heart Network was funded first by NHLBI about 10 years ago. We just started our 11th year of funding. So the mission of the Pediatric Heart Network is to conduct clinical trials and other clinical studies, of which we do several at a time, to advance the evidence base for children with cardiovascular conditions. We do this through 9 main clinical centers. We have auxiliary sites that help us with one or more of our trials. The New England Research Institutes in Massachusetts is our data coordinating center, and NHLBI is a significant scientific as well as administrative partner in this activity. Since September of 2001, when we were first funded, the network has launched and conducted 9 studies, 3 of which are ongoing. This includes a prior rarity in congenital heart disease, 4 randomized clinical trials. I'm going to talk about the single ventricle reconstruction trial in a moment. We also have established ongoing cohorts of both our surgical cohort and the Fontan cohort, which is now into young adulthood. So the thing that causes children to be moribund today most often is hypoplastic left heart syndrome. And again, although there have been a number of advances in recent years, still the morbidity and mortality is unacceptably high. And surgical innovation continues, which is fortunate. So the Pediatric Heart Network conducted a clinical trial comparing these 2 strategies. On the left, you see the Norwood procedure, which is the initial palliative procedure for hypoplastic left heart syndrome, with the enduring Blaylock-Taussig shunt from the first blue baby operation, providing the source of pulmonary blood flow. And on the left, the newer procedure, the RV to PA shunt, where the source of pulmonary blood flow is from a conduit from the right ventricular outflow tract to the pulmonary artery. And in about 3 years, we enrolled over 550 infants with hypoplastic left heart syndrome at 15 sites around the country and in Canada. The primary endpoint that the study was powered to observe was the rate of transplanter mortality at 12 months. And indeed, at 12 months, we found that there was a significant difference in outcome favoring the RV to PA shunt group. As with many other clinical trials, however, the story is evolving. So we looked at the primary endpoint here at 12 months, but when we examined data at the time of publication of the main results paper with all available follow-up, we found that the relationship no longer held. Small numbers, absolutely. But we're continuing to follow this cohort, and actually we've just started to look at the completed 3-year follow-up. So we expect that to be in press within 6 or 8 months. Now, to close this part of the story out, I remain encouraged that mainstream medical journals that primarily focus on adult research in adult populations recognize the value of pediatric research. This is actually the Pediatric Heart Network's second New England Journal publication. So again, with the theme of why do research in children? Well, they do have diseases that adults have, but with different pathophysiology is another reason. So I'm sure you're all familiar with the articles that started appearing in the early 1990s indicating survival and other benefits for adults with heart failure from ACE inhibitors. Pediatric cardiologists embrace this. Certainly children get heart failure, but for children with structural heart disease, the type of heart failure is not the same as in the trials done in adults of ACE inhibition. And one of the areas where there was a lot of use of ACE inhibition was in the single ventricle population, which was probably the most far removed from the trials in adults. So the Pediatric Heart Network decided to test this hypothesis. And we conducted a study where we enrolled infants with all types of single ventricle, not just hypoplastic left heart syndrome, randomized them in the first year of life to enalapril versus placebo on a background of standard care, and found that there was no benefit on any parameter measured, including the primary endpoint, which was somatic growth at a year, that there was no benefit of ACE inhibition. And in retrospect, this shouldn't be too surprising. The type of heart failure that children with single ventricle get just isn't the same. So although certainly it's understandable why people use hand-me-down drugs, it's not necessarily always a good idea scientifically. So another reason to do pediatric research is that hand-me-downs can be taken away. So this is a story about aprotenin in which Dr. Jonas is very interested. He wrote about it in an article published in Generic Medicine earlier this year. Aprotenin is the generic name for Translational. The FDA approved the use of aprotenin or Translational in 1993 to reduce blood loss during coronary artery bypass grafting based on clinical trials. No comparable studies were done in children, of course, as is the case with most of the drugs we prescribe. But off-label use in congenital heart disease surgery was soon embraced because of the perceived benefits in controlling bleeding, especially in neonates and infants. Time passed, things went well for a while, and then in 2006, the New England Journal published this article by Dennis Mangiano and colleagues, which was a prospective observational study enrolling more than 4,000 subjects at 69 sites around the world. They looked at the use of aprotenin, tranexamic acid, Amicar, and no treatment in adults. Interestingly, they didn't enroll all eligible patients. They had a formula for what proportion of eligible patients at each site would be enrolled. And they reported that aprotenin was associated with a doubling of the risk of renal failure and a significant increase in the risk of MI and heart failure, as well as stroke. Well, this started to raise a few eyebrows. But of course, it wasn't a clinical trial. The clinical trial came along and again was published in the New England Journal 2 years later in 2008. The blood conservation using antifibrinolytics in a randomized trial, BART, study was done at several institutions in Canada. They enrolled more than 2,000 subjects undergoing high-risk cardiac surgery and randomized them to tranexamic acid, Amicar, or aprotenin. The mean age was 67. The primary outcome on which the study was powered was a risk of massive bleeding. Did they find any difference in the risk of massive bleeding? No. However, the Data and Safety Monitoring Board that was overseeing this study was concerned about the 30-day mortality and stopped the trial on that basis. So at 30 days, the relative risk of death in the aprotenin group as compared with the other two groups was 1.53, and the confidence intervals were significant. 6% of the aprotenin population compared to 4% of the subjects in the other two arms died. And on the right, you can see the Kaplan-Meier curve. The red line, that's the lower line, is the aprotenin group. And the comparison between aprotenin and each of the other two drugs was significant at the 0.05 or 0.06 level. Soon thereafter, we started to see ads like this. This is a law firm inviting people to come forward if they thought they'd been damaged by Translational. And not surprisingly, Bayer voluntarily withdrew aprotenin from the market in May of 2008. Well, under pressure from congenital heart surgeons and others, Health Canada, which is equivalent to the FDA here, convened two expert advisory panels in 2008 and 2009. These results have just become public a couple of months ago. There's a lot in these reports, but the striking things are here. They found that the most prominent issue was the exclusion of 137 patients from the analysis after randomization. This is a major no-no. Once you're randomized, you're in the trial. And what was even more interesting is that the trend in mortality for the patients they left out was in the opposite direction of the trend in mortality for the patients that were included. And in the understatement of the century, they went on to say, this was not satisfactorily explained. So to go back to the Kaplan-Meier curve, they're barely hanging on to statistical significance by their teeth. If you put in 137 patients who lived instead of died, the significance goes away. I think in light of this, most people think it's extraordinary that this was published. Then Health Canada went on to say in September of this year that the benefits of Translational aprotenin outweigh the risks, and Bayer can resume the marketing of Translational in Canada. Can Dr. Jonas have his aprotenin back now? No, sorry. He can't import it from Canada because it's not on the market here, and even in Canada, it's only available to hospitals doing CABG procedures. There's never been a pediatric indication. Is Bayer going to market this in the U.S. now? Maybe not. It's a generic drug, and their lawsuit to profit ratio in terms of expenses is probably not favorable. Ironically, to bring it back for use in children would require a clinical trial. Bayer is happy to support this, but if a trial is positive, there would be no guarantee that Bayer or anyone else would market the drug here. So why does this illustrate the importance of research in children? Well, there was never a pediatric indication for aprotenin, and so it was easy to take the drug off the market for everybody because the FDA didn't have to distinguish, or Bayer didn't have to distinguish between age groups and the risk. Then there were some fancy manipulations of the data. The legal community got involved, and it's children who lose. So a more encouraging story now about why we should do pediatric research is the potential for hand-me-ups. Pediatric research that could benefit adults. So I apologize, I left a citation off this in the next slide. This was published by Eric Olson's group at UT Southwestern in Science earlier this year. What they did was to amputate the left ventricular apex. You can see the arrows show, and down here in larger magnification, higher magnification, where the incision site was. They amputated in neonatal mice the left ventricular apex and then serially followed the cohorts of mice along and looked at them at various days up to day 21 and found that, in fact, the left ventricular apex regenerated. This is H&E staining of the myocardium, and what you see is not scar, as you would see in adults, but you see cardiomyocytes. And Dr. Olson gave a talk at the American Heart Association last week, I guess it was, and elaborated on this story a little bit more. They figured out that for the first roughly week of life in the mice, they retain the ability to regenerate because cardiomyocytes are still actively in the cell cycle. Once they go out of the cell cycle and become more differentiated, then the response is what we see in adults, fibrosis and scar formation. But they have identified at least one transcription factor and another microRNA that may be able to reestablish the ability to regenerate. So just imagine if through research in juvenile animal models and development, we could come up with therapeutic targets that would lead to regeneration in adult MIs rather than the current scar formation. This would be hand-me-up science that would be paradigm shifting. And similarly, Dr. Vittorio Gallo here at Children's reported last week at the Society for Neuroscience on his work looking at oxygen deprivation in immature brains and its effect on myelination. We all know that premature infants have a lot of neurocognitive difficulties, and this is often due to white matter damage. So he and his team here have identified critical regulators of oligodendrocyte regeneration, which drives myelination, and thus could be potential therapeutic targets. So again, this would be great for preemies, but think about the potential for hand-me-up science in adults and others with stroke, multiple sclerosis, spinal cord injury, traumatic brain injury, and the like. My second to last reason to do pediatric research is that we really don't know what we're doing much of the time. So this is from the 17th century when bloodletting was common for fevers, colds, whatever. So a doctor at the time who didn't believe in bloodletting sort of advertised for this clinical trial. He wants to get 200 or 500 poor people with fevers. He's going to randomize them by casting lots. And so half of them will get bloodletting and half of them won't. And the primary endpoint is what the FDA would call a hard endpoint, funerals. There's no adjudication necessary. And we'll see what happens. And it turns out that, in fact, the people who did not get bloodletting had a much better outcome than those who did get bloodletting. But ironically, more than 200 years later, William Osler was still lamenting the inadequate use of bloodletting in medicine. So not only do we not know what we're doing, we're stubborn. And sometimes even with science, we don't want to change our opinions. You all know these modern examples of bloodletting. I'm not going to spend much time on this. Thalidomide, oxygen for premature infants. If some oxygen is good, more is better, right? No. And then bed rest for rheumatic fever. And I always like Welton Gersony's comment here that the notion that scientific advances sometimes occur getting rid of your old baggage. Okay, so now I'm going to spend a little bit of time on why more research isn't done in children. Well, pediatrics is actually a relatively young specialty. In fact, there was so much infant mortality early on that people didn't really feel that it was appropriate to devote resources to care for children. The American Academy of Pediatrics, founded in 1930 on the astonishing basis that children have special developmental needs and they're not miniature adults. Why isn't more research done in kids? They're vulnerable and we're afraid. The horrors of World War II resulted in the Nuremberg trials, and what emerged from that was the Nuremberg Code in 1949, setting forth principles governing human experimentation. The chief one, of course, was informed consent, and many people took that literally to mean that children couldn't provide informed consent. So for a long time, research really wasn't done in children. Then we all know about the Tuskegee syphilis story, but pediatric research has its own example, probably more examples, but at the Willowbrook School, which was in New York, this was a school for the mentally handicapped. It was built in 1947 to hold 4,000 children, and by the late 50s, early 60s, had 6,000 children. Parents desperate to have their children admitted in order to get the child in had to consent to have them involved in studies. The most notorious of the studies were the hepatitis studies, where children were intentionally infected with hepatitis in order to observe the patterns of hepatitis in an institutionalized population. When Bobby Kennedy visited the school in 1965, he referred to it as a snake pit, and it still took another 22 years for it to close its doors. So children are vulnerable, and people are afraid. Another reason more research isn't done in children is that it takes a lot of personnel. This is 3 Pediatric Heart Network coordinators measuring one single child. Even something as simple as how tall are you is difficult in pediatrics. In adults, they're not quite as, in adult studies, they're not quite as squirmy and wiggly. It doesn't take as many people. As we said before, children don't take pills. So developing liquid formulations of medicines is both expensive and time-consuming and uncertain. The Pediatric Heart Network spent $100,000 in a year on a formulation, an official formulation of liquid atenolol, which we all use all the time, which the FDA still didn't accept. Children don't vote. They're not a big market for drug companies. They're usually healthy. And another reason more research isn't done in kids is because today we don't see encouraging headlines like this. Instead, we see headlines like this, which are far removed from scientific inquiry. Another reason more research isn't done in children is that they're a diverse population. If you're a researcher working on adults, you don't have to worry about having people in your study who weigh 5 kilos and 50 kilos and undergo different developmental stages. In recognition of this, Congress enacted the Best Pharmaceuticals for Children Act in 2002 and renewed it in 2007. The purpose of this is to provide drug companies with incentives in the form of extension of patent exclusivity for testing drugs in children. And actually, this has been reasonably effective. Probably close to 200 drugs now have received exclusivity, and we've obtained a lot of useful information. For example, it turns out that some younger children, because of differences in developmental handling of the drugs, actually require larger doses than you would imagine rather than smaller doses. The latest development in this story is the funding of the Pediatric Trial Network, which is funded by Child Health at the Duke Clinical Research Institute. The purpose is to provide the infrastructure to conduct clinical trials for the BPCA's drug development program, and they are looking for collaborators if people are interested. I want to come back now to the Pediatric Heart Network. 2 years ago, the Pediatric Heart Network was joined by 2 other consortia, the Pediatric Cardiac Genomics Consortium and the Cardiovascular Development Consortium. The Cardiovascular Development Consortium consists of 4 academic institutions doing large-scale team science to unravel the persistent mysteries of the molecular underpinnings of the causes of congenital heart disease and genetic and genomic predictors of risk. The Pediatric Cardiac Genomics Consortium, which is led by John Kaltman, is 5 sites recruiting children and their families into studies to, again, look at genetic and genomic causes of congenital heart disease and predictors of risk. So together, these form the Bench to Bassinet program that Dr. Barul mentioned earlier, and we hope this will change the face of therapy for children with congenital heart disease by providing a continuum of translational care, translational research under one roof that can potentially identify therapeutic targets, genetic targets, and then test them in the Pediatric Heart Network. One of the things we learned in the Pediatric Heart Network was that another reason there's not a lot of pediatric research is that parents really don't have the resources to understand it. It's like learning a new language. You're randomized. Somebody's blinded. At the same time that they may be learning that their child has a critical heart disease, somebody's also approaching them about participating in research. So NHLBI, working with the Pediatric Heart Network's Data Coordinating Center, which actually has an award-winning media department, created this campaign to inform families. This is hosted on our website. It's accessible at the 7th grade level, and it's available in English and Spanish. And it consists of didactic information about stages of a study, but also in the center there's an award-winning documentary that involves experts in pediatric research, children and families who participated or not to provide a balanced picture in clinical research, and child actors. We've been fortunate to have support from the Child Health Institute, the National Center for Research Resources, the Gerber Foundation, and the National Marfan Foundation. And so now I'm going to close by letting the children themselves tell you why it's important to do pediatric research.
Me and my brother, Jackson, have a rare disease called chronic granulomatous.
I think it's ultra rare actually. It's a disease called Fabry disease.
It is something you're born with, and then usually the doctor texts you and says, "Ooh, there's something wrong," and then you just find out you have it, and then you just take a pill, and people try and make a way for maybe it's a go away or something.
FSGS doesn't really have a cure. So what we're doing now is we're trying to find out what medicines can help it control the scarring of the kidneys, and that's when they told me about the study.
So, you've been asked to enroll your child in a clinical study? That's scary. But you know what's scarier?
Until recently, and even still not very often,
very little testing has been done on kids of the drugs kids take, or might have to take someday.
So, yeah, it's scary. But we're taking stuff that's only been tested on adults.
That's pretty scary too.
Actually, it has affected a lot of stuff. I've missed some stuff, but I've been willing to do it because I think it would be good if I kept going. And it also helps me a lot, and it's going to be worth it in the long run because I'm helping myself and future generations and people who have the disease now. So if we can get this medicine approved fully and so it's open to everybody, that'd be great.
So thank you. I'd like to thank the Pediatric Heart Network team at NHLBI, including John Kaltman and Kristen Burns, who are here in the audience. And thank you.
So, Dr. Pearson, I just wanted to thank you so much for coming in for this very interesting and informative talk, and to say how proud I am of you as a Children's National Residency graduate.
Thank you.
And the forum is now open for questions.
The Nuremberg laws came out in 1949. The polio trial was in the 50s. Were all those 1.7 million patients consented? What was the informed consent process?
There was some form of consent. What's interesting, the pilot studies for that vaccine consisted of Salk immunizing his family. This was a common approach back then. There were earlier studies done in 15,000 school children in the Pittsburgh area. I'm sure it isn't what, it wasn't the 20-page dense form we would see today. But I think there was public information and understanding.
Yeah, thanks for a wonderful talk. I'm Max Koppes. I have to disclose I'm an oncologist, so I understand the NCI. What's the rationale for having heart, lung, and blood together in one institute? It sounds to, and you probably have some ideas, but what does blood have to do with lungs and heart? I know that blood flows to the heart, so you've talked today about cardiology, but I know that the institute is very much involved in blood and lung. And would there be a benefit of adding or dividing this institute?
So it's always good to have these existential questions on the eve of a holiday. Actually, the second NIH institute founded was the Heart Institute in 1948, so it was the Heart Institute. And then lung and blood were added later, and it's a great question, but I don't know the politics of that. We also somehow have the Women's Health Initiative study and sleep disorders. So I will say that, well, of course, you know, blood flows through the heart and the lungs provide oxygen. It's all focused on the heart.
One question I have is that we often hear from some of our colleagues, surgical colleagues and interventional colleagues, how do you differentiate innovation from research?
I think, you know, another kind of existential question. I think this is not so easy. There have been groups that tried to put together guidelines for thinking about how to think about surgical research in particular. We really need both, and I think as long as we're responsible, there's no harm in innovation in either field. You know, it was from innovation with the RV to PA shunt that you know, some initial surgeries were done, and then, you know, we were able to take that into a trial. So I think that with appropriate discussion with families and the like, innovation is appropriate, and then we just need to test it.
My question is whether more resources should be or could be put into attacking mythology that is really affecting, you know, medical practice. Do you know whether there's an effort to do that on a more organized basis? I mean, the Institute of Medicine has made some efforts to separate, say, measles vaccine from causing autism, but I just think there maybe more resources could be put into attacking mythology.
So you raise a great point, and I think one of the things that NIH does poorly, although under Francis Collins, I think we're getting better at this, is talking with the public about the benefits of research and what research means. It's much easier for people to look at sensational stories and believe those. And, you know, if you see it on the web, it must be true, right? Because it's in print. I think in general, I don't know that attacking the mythology is a useful strategy. I think we've seen this in the last presidential campaign and with global warming. You know, people are going to believe what they want to believe, but I think continuing to educate people about the importance of research and what, you know, how to think about things in a structured way. And I think we need to do that in ways where we're not talking in scientific terms, but converting things into lay terms. And I think it's something we all struggle with and could get better at.
That with drug development, we're seeing enormous barriers to drug development, prolonging the time from conception to actually getting out into practice new drugs. On the other hand, we're seeing this sweeping change in interpretation of research such that new paradigms are being accepted immediately upon publication. For example, with the aprotinin studies, which were flawed by Mangano and others, as well as with the Vandenberg trials with tight glucose control, which are now looking like that might not be the approach in critical care in critically ill adults and children. So how do we reconcile the two? In other words, how are we able to break down some of the barriers for new device and new drug developments to get newer innovation to the clinical to the bedside sooner? And how do we, like the prior question asked, how do we get a little bit better evidence medicine review before we totally change paradigms and swing the pendulum in the opposite direction? So you may have heard that one of Dr. Collins' initiatives is to create a Center for Translational Research.
and which would sort of replace the current National Center for Research Resources. And the impetus behind this is to try to bring resources together to shorten the time or eliminate the so-called valley of death between the time when you identify a drug target and then you can bring it to the market. So I think there are systematic efforts at NIH to try to bring the resources together. Our own institute has a couple of these. The Bench to Bassinet program is an example of this, to think in new ways about coordinating the basic science, the translational research, and the clinical science. I don't think, I have the right answer for how soon is too soon to put a drug on the market. It almost depends on the disease. People argue, on the other hand, that it takes too long to get things through the regulatory process here, and that's why innovation is increasing in Europe and we're being left behind. So you raise a great point with a lot of complicated aspects. Clearly, I think in the last decade or 2 decades, we've moved into a realm of much more critical evaluation of clinical research, and that will help. But do you have an answer?
Getting back to the topic at hand, it's obvious from your talk about the value of pediatric research, and there has been some incentive by Congress that you mentioned with the drug companies. What else can be done to encourage and increase the number of good pediatric clinical research projects?
That's a great question. So, one, we need to train pediatric researchers. The clinical and translational science programs, which Children's Now has one, has a specific requirement in it that's in legislation to devote some of the funds to pediatrics. I think this is why I give talks like this, to get the message out. I'm hopeful that our website, the Children in Clinical Studies website, which now gets 11,000 hits a month. Okay, it's not Google, but that's not bad for an educational site. Will help inform families, and actually, because there's no intellectual property associated with that site, hospitals like this one can download it, play the video in their waiting rooms. So I think we need to think harder about using social media and other more contemporary techniques to just inform people about the benefits of research. I mentioned a while ago, I don't think NIH does this very well. When you hear something from NIH, it's about a specific study, but we don't have good PR about why research is important, why we should have polio pioneers. I mean, you couldn't imagine doing that study today. And, more than 9% of the population needs to know what NIH is. So I would, I guess, charge everyone in this room with picking up the baton and helping to spread the word.
Are there any additional questions? Dan, do you have one?
I can speak loudly. So I'm Dan Desal, one of the Pediatric Chief Residents. Thank you for finally telling the story of pediatric research. I think the merits are clear. I think it's an interesting time. We're at a crossroads where the science has never been so promising and cutting edge. But as someone who's about to embark on a pediatric fellowship and considering a career in research, frankly, I'm concerned about the funding issue. And you're right, we want to recruit our best and brightest, but can we be assured that the funding will be there? As you look at your crystal ball, I know Congress is trying to cut so much. What do you think is going to happen with funding for NIH?
So I think we're looking at some lean years. I think a lot of us got used to the doubling of the budget, which happened of the NIH budget, which happened several years ago. I think you are right to be concerned, but I wouldn't give up hope. NHLBI is funding grants kind of at the lowest level that it has since I've been there in nearly 15 years. Somehow in this country we need to regenerate a dialogue about the importance of discovery. Instead, Congress seems to focus on grants that they don't like individual grants, and they are, I think, throwing the baby out with the bathwater. People all over the country are concerned about the research pipeline and what the low success rate for grants right now means for the next generation. There certainly are other funding sources out there, and at least at NHLBI, I don't know what fellowship you're going into, but we're the only institute that gives a very good preferential pay line. This is how we decide which grants to pay for early stage investigators, people who are within 10 years of finishing their terminal training. So you should go into a heart, lung, or blood fellowship. Or sleep. Don't forget sleep. My guess is the pendulum will swing back the other way soon, but you know, I can't lobby, but others can.
I have one question. I fully understand that basic science research is extremely important and feeds into clinical research. But if you had your druthers and hands over the $3 billion NHLBI budget, would you reapportion the budget a little bit to give more for clinical trials? Because, as you know, they're extremely expensive and really hard to do in, you know, a good clinical trial.
So actually, about 85% of the research budget for NHLBI goes to investigator-initiated grants. So these are grants that investigators like yourselves submit using standard NIH mechanisms. So we don't control very much of that. We, you know, we can influence some of it by issuing program announcements indicating our interest in research in certain areas. Programs like the Pediatric Heart Network are things where that we initiate internally and issue requests for applications or requests for proposals if it's a contract for, and that's really what we control. I think instead where we're going with clinical trials, because as you say, they can be very expensive. Several of our large adult cardiovascular trials have cost well in excess of $100 million. We are encouraging people who are applying with that type of trial, and things of that magnitude have to undergo prior review before we'll even accept them. We're encouraging them to think of less expensive, more streamlined ways to do clinical trials than has been the norm. It doesn't need to cost $12,000 a patient to study stable angina, for example. There are good examples using other techniques like cluster randomization of sites to compare strategies, and certainly with the advent of electronic health records, there are going to be ways to cut the costs of clinical trials in that fashion as well. So, you know, rather than tinker with the budget, which is hard to tinker with because much of it simply relates to what people are applying for, we're trying to make more efficient use of every dollar that we get.
Thanks again, and I'd like to thank everyone for the thoughtful questioning and thoughtful answers. And in the spirit of thanks, would like to wish everyone a wonderful Thanksgiving.
Thank you.